welcome everyone back to the first meeting of the Wilson Forum for the fall of this year. And I'm particularly happy today to welcome Bob Langer to the forum as our leadoff hitter, I guess I should say, using the baseball vernacular. Bob is an institute professor at MIT. He's a distinguished and celebrated educator, researcher, innovator, and entrepreneur, and who has introduced engineering principles into medical research and medical practice for the benefit of people all over the world. One prominent example of this is captured in Bob's founding role in the COVID-19 vaccine maker, Moderna.
Well, thank you for having me. And so what I was going to do, I was going to start with a slide that I put together on innovation, at least for me, and then try to go over some things that will support that. So maybe we'll just put that up
Okay.
And it's sort of 6 points that sort of go through my talk. It's on the next slide.
Okay.
So, and these are sometimes things that I often tell my students. First, to dream big, do things you hope will have a very big impact as compared to incremental research. You know, a lot of times when people leave the lab and they're going, you know, they want sort of security, even if they're professors, you know, and they think, well, you know, we'll publish some extension of what we've done. And my advice is not to do that. It's to do things that hopefully will have a big impact. It's a riskier approach, but I think in the end it pays off. The second point is that conventional wisdom isn't always correct. That was already alluded to when we were talking about, you know, one of the gentlemen was talking about some of my research, and I'll go over that. The third is, for me at least, was exposure to very, very different disciplines. The fourth is taking risks and the fact that failure is okay. I don't like it, but I think you have to, if you try to do things that'll have a big impact, you're certainly gonna fail. I've failed plenty of times. Fifth is that if your idea is important, expect lots of criticism. And six is not giving up, at least almost not giving up. So let me try to back some of those things up, but also in so doing, tell you a little bit about my own career. So I got my doctorate at MIT in chemical engineering in 1974. And when I got done, pretty much all my classmates went to work in the petrochemical industry. And so I thought I'd do that too. And I applied to these companies, and I actually got 20 job offers, 4 from Exxon alone. And believe me, I wasn't that good. They just gave lots and lots of offers. I could tell lots of stories about that. But at any rate, I really wasn't excited about those. I still remember one of them saying that if I could just increase the yield of one chemical by a tiny percentage, how much money that would be worth. And I just remember flying back to Boston thinking I didn't want to spend my life doing that. And so it made me think, what did I want to do? And that I've always told is, you know, those interviews were very helpful to me in thinking about, you know, what I didn't want to do. So one of the things I did when I was a graduate student and I was supposed to be doing my thesis is I helped start a school for disadvantaged high school kids called the Group School. I got very involved in developing new chemistry curricula, math too. But one day I saw an ad to be assistant professor at City College of New York, to do that, to develop new chemistry curricula there. And I was very excited. I wrote them a letter applying for a job, but they didn't write me back. But I liked the idea, so I found all the ads I could to do something like that. I wrote to maybe 40 different colleges, and a lot of them not particularly good ones. But anyhow, none of them, I don't think any of them wrote me back. So that wasn't going so well. But again, I thought, well, what I do want to do is use my chemical engineering education to help people. So I thought about medicine, and I wrote to a lot of hospitals and medical schools, and they didn't write me back either. And then one day I was walking into the lab. I was getting pretty discouraged by this time. And one of the older scientists said to me, he said, Bob, there's a surgeon in Boston named Judah Folkman. And he said, sometimes he hires unusual people. Dr. Folkman had an idea, if we could go to the next slide, if we could go to the next slide. Is it showing?
Oh, it should.
It hasn't switched yet.
Yeah, here we go.
Oh,
Okay.
sorry.
Now, yeah, so his idea was that if you could stop blood vessels from growing, you could stop cancer. More particularly, and this is sort of just a slide showing how tumors grow in relation to their blood vessels. And the important thing is that the blood vessels give the nutrition to the tumor that enables them to grow. And ultimately, if you go to the top picture, you see they're very vascularized. And of course, the tumor spreads through those blood vessels too. That's a process called metastasis, which often kills. So he had a theory that the way this all happened was that the tumor secreted a chemical signal called TAF, tumor angiogenesis factor. And that if you could somehow stop that, achieve anti-angiogenesis, that might be a whole new way of treating cancer. So I thought, I don't know anything about biology. I mean, the last time I have taken a biology course was in 10th grade, but I thought it was a really good idea. It turns out he was widely criticized for this. This is 1974. It turns out he's also right, but this is a point of conventional wisdom that I was mentioning. And so he asked me, he said, well, he offered me a job. It was by far the lowest paying job of any of the jobs that I got offered. But he offered me a job to do two things. He said, one, prove he was right, and in so doing, isolate the very first substances that might stop blood vessels from growing. One of the key things, and this is why we're showing this next slide that you see here, is that one of the key things is that you have to have a way to study blood vessels, and there wasn't one. So we had to invent one. And the way we did it is we could put certain tumors in the eyes of animals and like rabbits, and then we needed a tiny delivery system like a microparticle or a nanoparticle that could deliver large molecules. All the molecules we were isolating at that for blood vessel inhibition were large molecules. And the idea of doing drug delivery of large molecules or even small molecules at this point, 1974, was really very, very new. The only company in the world at this time that was really working on that was a company called Alza. They were delivering small molecules, and Dr. Folkman was on their board. And he asked they had very famous people on their board, Nobel Prize winners in chemistry and so forth, and he asked them, could they help us? But they all said, if we could go to the next slide, that you really couldn't do it. If we could go to the next slide, because the molecules were just too big. If you put them in a particle, they were just too big to get out any more than, like, if I asked, could any of you walk through a brick wall? I mean, the answer would probably be no also. But nonetheless, if we were going to solve the blood vessel problem, we had to solve this problem. So I spent 2 years in the laboratory, more kind of Edisonian-like, experimenting with different materials, different designs, and I actually found over 200 different ways to get this to not work. But on the next slide, we were able to make little microparticles or nanoparticles. You see a picture of one on the left, cut in half on the right. And we showed in a paper in Nature, next slide, in 1976, that you could use this to release molecules of any size. Also, and this will come up later, this was also the very, very first paper showing you could ever take tiny particles and use them to deliver nucleic acids. And for reference, DNA and RNA are nucleic acids. Now, I want to go back to 3 months before this paper came out. I got asked to give a talk at a major scientific meeting in Michigan. And it was really the first talk I'd ever given at a conference. And I practiced it for a long time. And I was 27 years old. I got up and I gave the talk. And I thought when I was done, I didn't do too badly because I didn't forget too much what I was going to say. I didn't stammer too much. So I thought all these older scientists, chemists and engineers being nice people, that they'd want to encourage me, this young guy. And yet when I got done, I stepped off the podium and a whole bunch of them came up to me and they said, we don't believe anything you just said. They basically said, if we go to the next slide, what the Alza people said, that the molecules were too big. And also I was using organic solvents like methylene chloride and ethanol to do the encapsulation. You almost always have to do that with synthetic polymers. And everyone said that those would destroy all these substances I was putting in. And actually things went downhill for me from there. If that were possible, I tried to get research grants. My first 9 were rejected. Generally I'd send them because I was doing health-related research to NIH, and so the reviewers said, well, Dr. Langer is an engineer, he doesn't know anything about biology and even less about oncology. So I didn't get any grants for a long time. Also, I really love being a postdoc with Dr. Folkman and being at Children's Hospital. But a lot of my friends said, to me, Bob, you probably shouldn't do that forever. And so I did start applying for faculty positions. And of course, since I'm a chemical engineer, I applied to chemical engineering departments. And no chemical engineering department in the country would offer me a job. They interviewed me sometimes, and I don't think I was a terrible interviewer, but basically they'd say to me that this biology stuff I was doing didn't make any sense. I should try to be doing energy or, other things that chemical engineers more normally do. So I didn't get any jobs. But Dr. Folkman introduced me to Nevin Scrimshaw. Nevin Scrimshaw was also on that ALSA board that I mentioned. He was head of the nutrition department. And Nevin Scrimshaw, if any of you had ever met him, he was, a giant, probably the world's leader in nutrition. But he was also, as a department head, a benevolent dictator kind of department head. So he liked me, so he offered me a job as an assistant professor, but he didn't ask anybody else in the department what they thought, which might have been okay, except for one thing, which was that the year after I joined the department, he left. And so a lot of the senior faculty decided to give me advice, and their advice is I ought to leave too. In fact, on the next slide, just to back that up, a few years ago, I was getting this award, and one of my colleagues at the time of this was all like about 1979, was Mike Marletta. Mike is a very famous professor today. He was head of Berkeley's chemistry department, head of Scripps, member of the National Academy of Science. He was giving us, he's a good friend. He was giving me a speech a few years ago, what it was like to be with me in 1979. Here's what he said. One evening, I went to a faculty dinner at a Chinese restaurant with me and some senior MIT professors. He said, a senior scientist sat quizzing us while smoking a cigar. He said, when the older scientist heard my concepts for drug delivery, he blew a cloud of smoke in my face and said, you better start looking for another job. So that was all kind of discouraging, but I made the point to you earlier about not quitting easily. So I kept my head down and kept trying. And one of the things, of course, that we did was I wanted to prove, what Dr. Folkman originally asked me to do. Could we actually isolate blood vessel inhibitors? And so I isolated maybe over 100. And I'm going to show you a very bloody slide in the next slide. Don't look if you don't like the sight of blood. But what we did is we probably did 2,000 rabbit eyes, over 100 fractions. Almost every one looks like what you see on the left. You get a sheet of blood vessels growing to the tumor, and the tumor will eventually grow because of those blood vessels. It would actually kill the animals they're sacrificed before this. But one of the fractions happens to do what you see on the right, where the vessels are lower, they're sparser, and they avoid the tumor. And this really showed for the first time ever that blood vessel inhibitors did exist, and would provide bioassays by which companies could and academic scientists could isolate all future inhibitors. Now, this is a 1976 paper. Probably any of you that work in the medical field know just how slow it is to go from a basic discovery like this to a real product that can help people. Here, it took 28 years from this paper, if we go to the next slide, before the first blood vessel inhibitor would get approved by the FDA. And that was Avastin. Avastin was, Bob Swanson was mentioned before. He asked me to consult for Genentech. I don't give myself much credit for they had people like Napoleon Ferrar and others that did great work. But at any rate, Genentech putting billions of dollars in and using some of the techniques that we had developed, I got the first one approved, that which is Avastin. That's probably the number 2 or 3 best-selling biotech drug in history. And what you see is this would continue to today. I mean, even last year, 5 angiogenesis inhibitors were approved. And they're not just approved for cancer, they're approved for many cancers. In fact, Nature, the journal, estimates that 500 million people will use these. But they're also approved now, and really the only ones that are approved for diseases of the back of the eye, the blindness in the back of the eye, which is macular degeneration and diabetic retinopathy. And these are all blockbuster drugs, multi-billion-dollar a year drugs that have had a giant impact on people's health. That came from really the research Dr. Folkman started, and that I was fortunate enough to help with. Another thing that Dr. Folkman said to me one day when I was working there is that we should file a patent. So as Leader Nelson was mentioned before, I have quite a few patents, but then I had zero, and Dr. Folkman and Children's Hospital had zero too. So Dr. Folkman said, well, Bob, we should file a patent on the delivery stuff. So we did, and 5 years in a row it got rejected. And the head of the tech transfer office at Children's Hospital was a guy named Al Brazaghini. And I remember he came to see me one day after the 5 rejections. He said, Bob, you're just wasting so much money. This is never going to get allowed. He said, you should just give up. But I don't like to give up. And science wasn't working, obviously, when the lawyers tried to explain it to the patent examiner. So as I told you in the beginning, when I first started doing this, people said to Dr. Folkman and myself it was impossible, it could never work. So I wondered maybe someone had written that down somewhere. So I did what's called the science citation search. I did it looking back at our Nature paper to see who cited it. And I did this now 6 years later. And I found actually a couple of very good papers, but one in particular, if we could go to the next slide, that I was going to mention. And so this was by 5 very famous material scientists. And I had no idea it was written, but they're describing this field. They said generally the agent to be released is a relatively small molecule with a molecular weight no larger than a few hundred. One would not expect that large molecules could be released because of their extremely small permeation rates. However, Folkman and myself have reported some surprising results that clearly demonstrate the opposite. So I thought, boy, that surprising word, that's really good. I showed that to Dr. Brazaghini, and he flew down to Washington to show it to the examiner. And the examiner said, gee, he had no idea. He said, tell you what, he will allow the patent if I could get written affidavits from each of these 5 people that they really wrote this. It's a true story. So I wrote them, and I was an assistant professor, and they were all nice enough to write me back they really wrote it. And we got a really broad patent. Next slide. And with that patent, actually, it turned out I got asked by a couple multi-billion-dollar companies to one in animal health, one in human health, to help them. And I mentioned the patent, and they licensed it from the hospital. And they gave me a grant and a consulting fee. And I was very excited that they were going to work on this. And they did. They worked on it for about a year, but then they just kind of gave up. And then Alex Klebanoff, one of my friends, said to me, he said, Bob, we should start our own company. So we did. And I also started trying to help other companies, and Children's would do some licenses to them. And because of some of those companies, Alchemies being one, but Takeda being another, let me just go to the next slide. And what happens is now there's all kinds of products based on this and other inventions that we made. Everything from Lupron Depot, which Takeda made, which was a $3 billion a year product that and basically it's a peptide that's used for treating prostate cancer and endometriosis. And people tried to deliver it orally and nasally, but it couldn't, it was too big. And when they injected it, it got destroyed right away. So it was put in little microspheres, and it was, still widely used today. And many others, drug-eluting stents, that's based on another one of our patents that Angiotech copied, and Gliadel wafer is another one. And Vivitrol is Alchemies, which I mentioned. That's one of the major ways of treating opioid addiction. And actually, even and again, a lot of this, you can't, because patents only last so long, can't be based on our patents, but they basically use the identical technology. One of the interesting ones is actually in the bottom right, which was an area I had no idea about, by Yanni Zohar, who's an expert in aquaculture. Basically, he said, well, can we use this to control fish spawning and even preserve rare fish. And BARD, the Israeli American Granting Association, basically said that this was probably was the most important grant they ever gave, and was largely responsible for this now $250 billion aquaculture industry, and even enables you to preserve rare fish. So anyhow, these got used for a lot of things. But most of them are implants, they're microparticles, and you could do all that by that Nature paper I wrote or extensions of it. But still, a lot of times when you make things really small, you run into problems, like you get aggregation because it's like wet sand. And sometimes there are these cells called macrophages, and they eat foreign particles. So what we did with Vladimir Torchelin and others, next slide, is come up with a disguise method by putting, like if any of you ever took Miralax, that's polyethylene glycol, and we made coatings on it and published this also in Science. Let me just show you a video which illustrates this much better than what than I can do on the next slide. This is from Nova, the TV show that came to our lab. If we could turn it on. Is there sound? There should be sound. Did the sound come on? Oh, there's no sound?
No sound.
Nope.
No sound.
Okay, there should be. But anyhow, what I was going to, basically, it just describes better than what I did. But the important thing is it's you have this coating, and then it goes to the cell. It doesn't really blow the cell up quite like they showed in the in this.
You know what, Bob, once again, I heard the sound perfectly. Can we try that one more time? Because I thought it was pretty informative. Can we do
Yeah,
that?
no, they did a good job, better than I will do.
Okay. Oops, wrong one. Sorry. Oh, I'm getting ahead of myself. Sorry.
He starts with a nanoparticle of anti-cancer drug. That gets encased in a plastic that releases the drug over time. That in turn gets a special wrapping that disguises the package as a water molecule to fool the body's immune system. And last but not least, the address where it should be delivered, a key that will only fit the lock of cancer cells.
And it should also make a big noise when it blows up the cell. But maybe it didn't do that. At any rate,
I hope everyone heard it.
yeah. But at any
Yes,
rate,
yes.
it doesn't really blow the cell up. But what I was going to say is, so that would help be that ends up being one of the key components in nanoparticles, whether you're doing it for polymers or lipids. We've also done work, I didn't talk about that, on lipid nanoparticles probably for the last 35 years, and developed other principles for them too. At any rate, that's going to come up next. So as I mentioned, so one of the things that I learned was, like working with large companies, a lot of times, I mean, even though I loved getting grants from them and helping them consult, is they wouldn't take new ideas. So I got involved in starting quite a few companies, a lot with the technology licensing office that Lita headed for many years. And so one of them, and this wasn't through an MIT patent. By that time, all our patents had expired in these areas. But the one that, but we developed all this kind of core technology of creating nanoparticles, making ones with PEG and so forth. So actually in 2010, if we could go to the next slide, Derek Rossi came over to see me. He had done work on modifying messenger RNA for stem cell work, and was interested in starting a company. And the idea we thought about, along with Nubar Fayan, who's an MIT graduate and started Flagship Pioneering, and Ken Chen, who's a cardiologist, is shown here, that maybe you could make a company to make messenger RNA therapeutics, put it in nanoparticles that you have to do because you have to protect it for, otherwise it'll get destroyed. And you could make protein. By this time, I had been an advisor to Genentech, Bob Swanson's company, for over 30 years, and I could see what they had done with protein therapeutics. It really changed the world. But but it takes a long time to make proteins. It's very expensive. As you all know, if you try to do a vaccine, say flu vaccine, you have to make eggs. You have to guess a year in advance even what the vaccine is going to be against, like H1N1, H2, whatever. So you're making a guess. Here, you could do this, we thought, incredibly fast, because if you could give the messenger RNA to the patient, the patient would make the vaccine. You wouldn't need a factory. You wouldn't need eggs. So, and you could also, as shown on the next slide, you could do things you could never do before, make different membrane-bound or intracellular proteins. So we started Moderna in 2010, and Moderna, really was widely criticized. I mean, the press, stock analysts, even journals like Nature interviewed scientists, and everyone said it was going to be like, or not everyone, but a lot of people said it was going to be like the next Theranos. But the scientists at Moderna, I thought, were great. Number of them were my own students. And by the end of 2019, in spite of that criticism, we had 13 products in clinical trials, early stage, but in clinical trials, 8 of which were vaccines. And then, of course, as you know, COVID came. And I want to show you on the next slide what the Moderna scientists did. The Chinese scientists published the virus genetic sequence of the spike protein, on the virus on January 11th. It took just 2 days for the Moderna scientists to create this vaccine, and then put it in nanoparticles, ship it to our clinical partner, NIH, for testing. And then what you do is you do a phase 1 trial. I'll go over that in a second, which is try to look at the highest safe dose in humans. And that started in March 16th. So mostly what you're trying to do when you do this clinical trial, what's called the phase 1 trial, is see if it's safe and see if you get antibodies. And we had that data in 2 months later. And of course, you're a public company, you have to announce it. And so Moderna announced it. And I thought, finally, the press and scientists will, cheer, because this could really help a lot of people. However, let me show you the front page of our local newspaper, the Boston Globe, on when this was announced. So here is the headlines when we announced it. And the headline is, "This is not how you do science." And they put my picture on the front page. So that was a little discouraging, but again, you keep trying. And what happens is that, after you do a phase 1 trial, you do a phase 2 trial, and then you do a phase 3 trial. And the phase 3 trial in this case was 30,000 patients. It's a double-blind trial. 15,000 get a placebo, 15,000 get the vaccine. And I'm on the board. I've been on the board of trustees board of directors of Moderna since I helped start it in the beginning, still on it. And when we announced it, when they broke the code in November, this was just 6 months later, what happened was, as people may know, it was on TV and everything. And finally, people did cheer, because it was not a single person died in that 15,000, not a single person went to the hospital, and it was 94% effective. Just to go over some of the things about it, the next slide is just a vial. This has been used all over the world. There's only 2 vaccines that have, for the longest time, that ended up being approved, and they're both messenger RNA and nanoparticles. This is for COVID, but now there's a third. But they've had an enormous clinical impact. The next slide just shows that CDC data, that if you compare unvaccinated to vaccinated and no booster, or vaccinated with a booster, the enormous differences. And if you look at effectiveness, like I said, next slide, it's 94%. Pfizer and BioNTech, which is also quite good, 91%. By the way, just for reference, flu vaccine if you generally look at that, those are usually 40 to 50% effective. I never realized that myself till I got flu. I thought, gee, if I get a flu shot, I won't get flu. And then I remember the doctor telling me it's only 40% effective. But that's still worth doing. But at any rate, that was, you know, so these are really very, very high numbers in terms of effectiveness. And of course, it just is at the tip of the iceberg of what I think you may someday see. And just to pick one other thing that Moderna and Pfizer, Moderna in this case Merck, have done together, next slide, is the idea of personalized cancer vaccines. And this is an idea that I think really could hopefully someday change cancer research. So this trial was also announced, public information, used 157 patients with advanced melanoma. And the idea is that you're going to create a vaccine just for that individual. So that individual will have different mutations. Each individual may have different mutations. And you take up to 34 of these and you incorporate the genetic code into the vaccine, just exactly the way we did with COVID, mRNAs and nanoparticles. And what was announced is you have this randomized trial, Keytruda, that's Merck's drug. It's an immunotherapy drug. It's probably the best drug out there. Half of them get Keytruda, half got Keytruda plus the personalized cancer vaccine. And the risk of recurrence or death was reduced by 44%. And a month or two ago, they also announced that in the same trial, the reduction in terms of metastasis was 65%. So this was also very encouraging. And basically, if you look at the literature today, in spite of all the criticism, you know, that I got 50 years ago, the next slide just shows that paper in Nature, really, when they talk about me and Folkman, that was a paper that, you know, first enabled one to encapsulate these, you know, nucleic acids, and considering someday that you could create DNA or RNA as a drug. Now, that's where we are today. I'd like to go over a little bit about where we are tomorrow on this. I was visited about 12 years ago by a software entrepreneur, and he has been very interested in helping people in the developing world. And he asked me if we could, well, I'll just show you a picture. He came to my office on the next slide, and that's Bill Gates, and on the right, and me on the left. And he said, well, Bob, if we could go to the next slide, you know, he said, people, and this was way before COVID, he said, people in Africa, they don't come back for a second injection. He said, could you ever create a way where you could give one injection and you'd get pulses at various times? So if you look at the middle bottom, I'll show you our idea. The red dot is the vaccine, but we're going to make what I'll call self-boosting vaccines. And so that red square is covered by a gray square. And what we thought about is maybe think of it as an eggshell. And we can make that by changing the polymer composition, changing that eggshell so it can break at different times. And what we did, next slide, this was done by Anna Jakulenik, is that in our lab, is that by taking polylactic glycolic acid, that's FDA approved degradable polymer that actually breaks up into water and CO2, that we could make it break at pretty much any time by changing the ratio of lactic glycolic acid and the molecular weight. And so that's what she did, and you could get bursts at any time. And what we hope to someday do, and we've been working with Mark Prausnitz, who's another one of my former graduate students, who's now a professor at Georgia Tech, and the Gates Foundation has supported this too, is next slide, is that someday what we hope to do is put these in microneedle patches. You see a close-up of that on the right. And the beauty of microneedles is they don't go in very deep, so they don't hurt hardly at all, but they go in deep enough that you can give the vaccine. And so what you see on the left is they look like a little Band-Aid, and you put them on in the center, and you just put them on the patient, and it'll give you the vaccine. Someday we hope to ship these all over the world. In fact, to that end, BARDA contacted Anna, who I mentioned in our lab several years ago, and they said, well, could we even make portable 3D printers that could make these things? If we could go to the next slide. This actually just came out a couple of months ago in Nature Biotech. John Aristotle's the first author. And so our team basically made a tiny little tabletop 3D printer to make these microneedles. You see pictures of them here. And what's really exciting about this, next slide, is that they actually work just as well as the microneedle is. This is against the receptor binding domain of the spike protein. But if you look at the microneedle printer, the MNP, that does just as well as the intramuscular injection. And what was also great is it was more stable actually on the shelf over 6 months because it's in a drier state. So we hope that someday we'll be able to help people in the developing world like this. Let me just go over a few other areas that we've worked in. And another one, which Ram Sasaskaran came to, and Ganesh Venakatraman, if we could go to the next slide, they were both students and postdocs in our lab. And one of the things we wanted to do was do some work on polysaccharides, which are more complicated than even proteins or nucleic acids, because they're polydispersed, meaning they're a mixture. And they also had done work in our lab on cloning enzymes on this and things like that. And we decided, next slide, to try to create a glycomics company rather than a genomics company. And in particular, the idea is, since you have these mixtures, you're actually injecting a mixture like heparin. I don't know if any of you have taken heparin. Heparin's a mixture. That's a polysaccharide, but it's not just one molecule, it's many molecules. And so our idea, next slide, was that you could fractionate these things like heparin to find what we call the right glycoform. But what we did is we developed the first sequencing approaches to create these complex polysaccharides. And it uses, for the scientists in the audience, 3 things: molecular scissors, those are the enzymes, that's which cuts these in various places, and then mass spec that tells you the sequences of the cuts. And then Ram and Ganesh developed an algorithm to put those cuts back together. So for the first time, you could get polysaccharide sequences. And we started another company with them, Momenta, next slide, and to make heparins, other complex polymers, and new glycoproteins. Next slide. So this is just Momenta that we started, and we actually were able to, in 2000, we got venture capital, and Alan Crane came to be the initial CEO, and then Craig Wheeler, and they did a terrific job. And we got a number of drugs approved, including low molecular weight heparin, which is Levinox, which was the first complex drug approved based on analytic data, and actually the largest syringe launch in history. And Momenta was acquired by Johnson & Johnson in 2020. One of the other things that happened, next slide, is that you could make, this is the enoxaparin, but another thing that happened, as some people may remember, maybe not, was that 15 years ago, there was this terrible heparin crisis. And the next slide just shows some of the headlines that that people found problems with this, with contaminated heparin, and it led to deaths in many different countries. So what we did is Ram and the people at Momenta, and actually the CDC and the FDA, we all worked together to help solve this problem. And we did. The next slide shows that in Nature Biotech in 2008, we found the contaminant. It was an oversulfated sulfate. And then again, 2 articles in the New England Journal of Medicine show, again with the FDA and CDC, showing, and after this, there were no more deaths. I thought I'd go over just 1 or 2 other ideas. And another idea that I had along with Jay Vacanti is shown on the next slide. And that is that maybe you could someday make tissues and organs from scratch. Again, this is going to use materials. But our theory, and this is from a paper we wrote in Science many years ago, it's been cited about 9,000 times, was that you could basically use almost any cell type, but if you injected it at random, then what would happen is not much. but if you put the cells close enough together, they could actually reform tissue structures. So that's what we did. And the idea is that you could make almost any new tissue this way. And one of the things that this led to was artificial skin that was licensed by a couple companies. And I'll just show you an example of a baby that was treated this way. It's now FDA approved. Next slide. So here's a little baby very badly burned. You can take the product, next slide. And that's a neonatal fibroblast on the scaffold. You can put it on the child, next slide. And if you come back 3 weeks later, next slide, and a month later, 6 months later, he's pretty much healed. And this has now been approved for patients with diabetic skin ulcers and burns. You could also, we thought, make organs and tissues on a chip. And Gordana Vunyak and Milika Ritzik in our lab, what Gordana is now Columbia, Milika now at Toronto, they actually made a heart on a chip. And the next slide just illustrates that. You can take heart muscle cells and put them on, and they'll get you this characteristic structure. And Lisa Freed, when she was working with it, even took videos of these, and what she found, and she was involved in this whole thing too, is shown on the next video. And what you see is literally 6 million heart cells just beating as one. And you can take an EKG of these as shown on the next slide. And Milika and Gordana started a company to do drug testing on this, which has worked with about 30 companies. And I just thought I'd show you on the next slide that there are a whole bunch of engineered tissues that we're working on. I really won't go through these much other than to illustrate, mention them: cartilage, spinal cord, GI tract. Some of them are on a chip, like you see this brain on a chip that we're working on with Li Wei Sai at MIT. This is being done by Alice Stanton, a postdoc in our lab. Blood vessels, that's led to Humacyte, which Laura Nicholson, one of our fellows, started, and many others. So I thought I'd end with something very, very different. Almost everything is that I've talked about is medicine. But, one day, one of my former graduate students, Amir Nishat, came to see me, and he said, well, Bob, he's at Polaris. Polaris and Flagship have been the two biggest supporters of the things that we've started. But Amir said to me, he said, Bob, we've given you all this money to start, medical companies. He said, we want to start a company in cosmetics and hair care. And so I said, well, what do you have in mind? If we could go to the next slide. So he said, well, we've been talking to a lot of hairstylists, and they tell us frizz is a really big problem. And maybe we could come up with better ways to do that. The next slide just shows you this company we started called Living Proof. And, when I started talking to the hairstylists, and Dan Anderson, who was also one of my postdocs, and now is a full professor at MIT, he was also very involved. I got him involved in it too. And what we found was interesting, that even though this was started in 2005, that even though there were a lot of anti-frizz products, they all did pretty much the same thing. The next slide shows that basically they all use pretty much silicones, and the idea is that they'd formulate them differently, maybe different, different smelling substances or others, but basically they all use pretty much silicones. And we took just a more fundamental look at this. The next slide just gets to the, more mechanistic, not that this is that deep, but basically what causes frizz? Well, it's humidity. And if you really look at silicones, and you look at just on the next slide, they really don't fight humidity all that well. You just compare, an isotherm, and you see that it doesn't do much better, a little bit better than water maybe, but not very much. So what Dan and some of the other people, Betty Yu, she was another one of my students that Amir and us recruited from ALZA, basically said, well, are there any FDA approved products that are very hydrophobic? Well, first I should take a step back. Silicone is somewhat hydrophobic, but if I asked all of you what's the most hydrophobic thing you could think of, what would you say? Well, what we said was a Teflon frying pan. So what we did is we said, well, what's the chemistry of that? And it turns out that it's a polyfluoroester. And what we asked then is, were there any things like that that were approved by the FDA? We're not going to put a silicone frying pan in your hair, but maybe there would be similar molecules that were approved by the FDA. And it turns out that there are some that were approved for use for coating intraocular lenses. So we looked at that, and actually the company patented that. And the next slide just shows what that is. And it worked a heck of a lot better than silicone. And so what it did, next slide, is that led to a whole bunch of products that people use. And I thought I'd show you a video. I hope this works on the sound that was done.
Today's leading edge of invention,
Can we turn it up?
a complete no-brainer. MIT.
This is channel 5.
New kinds of implants to treat brain cancer, to treat different kinds of cancer and other diseases.
Bob Langer and Dan Anderson run one of the biggest academic research labs in the country. Langer alone has over 750 medical patents, more than anyone in the world. He and Anderson have had amazing success inventing novel medical materials and life-saving therapeutics.
Well, certainly it's one of the reasons I really like my job. I mean, you really feel like the work you're doing on a day-to-day is not only scientifically interesting, but also can have a real impact on people's lives.
Now, their work on stem cells and damaged spinal cords has had to make way for another vexing problem. Bad hair days. The result? No frizz. Based on Anderson and Langer's science, no frizz has taken the beauty world by storm.
It's so amazing that the big companies haven't been able to solve these problems in such a short time. We've been able to really tackle problems that have really dramatically affected women for many years.
Rob Robillard is CEO of Living Proof, makers of No Frizz. This Cambridge startup secret weapon, science. Using renowned researchers from outside the beauty industry to apply new thinking to age-old problems.
This side of her head was treated with one of those silicone products. This side was styled with no frizz technology.
Okay, we found the holy grail for frizz. It's amazing.
Noted stylist Mitch DeRosa is a co-founder of Living Proof. He says until now, the fight against frizz has relied on silicone-based products, which keep moisture out, but can leave hair heavy and greasy. Along come Anderson and Langer and their discovery of polyfluoroester.
This creates a weightless shield sheathed around each hair fiber.
For Langer and Anderson, there's nothing trivial about their work for a beauty product. After all, they consider themselves equal opportunity problem solvers. Do your fellow professors teach you sometimes about the frizzies, or they have a fun with it, or they?
A lot of them use it.
Yeah, so anyhow, so that was one product. And then a second thing that they asked us was, could you give hair more body? And, Dan and Dan Anderson, we actually have these patents, in fact, again, in MIT's technology licensing office that Lita ran, we actually created a whole family of polymers, polybeta amino esters, libraries. So we had composition of matter patents, and the TLO licensed them to different companies for like gene therapy reagents. But anyhow, they licensed them to Living Proof for, we found that they could actually, one of them could give more body like this. So that leads on the next slide to what we call full. Now, one of the things I learned, unfortunately, when you do medical stuff, usually all you need is, I shouldn't say all you need, but you want good data. Here, I remember one day we hired a woman who was for vice president of marketing, Grace Ray. Later, she became CEO. But we hired her, and she said to me one day, Bob, she said, 95% of the people who said people like this. They said 95% of the people who use this reuse it, and 95% like it. She said, but way less than 1% even know it exists. So they said, well, we need to actually get somebody who's much more well known than you, that that the world might listen to. So what they did, next slide, is they got Jennifer Aniston involved. Her with me, and then of course the company started doing extremely well, and actually Unilever eventually acquired it for quite a bit of money. And I just thought I'd show you one last slide too, which Betty Yu, who was one of my grad students, also made. So Unilever just bought the hair care part, but we also came up with a way to get rid of wrinkles. And that became a spinoff that Shiseido acquired. This was actually published in Nature Materials, but it's a little cream. We did this with Rox Anderson, Betty Yu, and that you could put on and get rid of wrinkles. And I think it's now being sold in Japan by Shiseido. I know they also acquired it. So anyhow, I thought I'd just end with that, just for the non-medical people in the audience. But it's been a pleasure to speak to you, and happy to try to answer any questions.
Bob, I want to thank you for that, and just to make I thought the comment about equal opportunity problem solvers related to living proof. I just want you to see that I'm one of your users. I use this every day.
Well, I know the reason I don't usually put it in the talk, but when you wrote me the note, you mentioned that. So I thought, well, probably I should put it in the talk, and it's fun. I mean, really, it's amazing. I've been to different places all over the world, in Singapore, Sweden, somebody comes and meets me and pick me up, and they say, well, I used, your shampoo today, just like you said.
Yeah. Well, I think the slogan that accompanies living proof just says it all. This, as I remember, the slogan is, "We are the chemistry, you are the living proof." And I think that really captures the whole spirit of this.
Yeah, they had great people. We had great people working there. It was a lot of fun to do, and a lot of times when people come to my office, I said, well, Jennifer Aniston sat there or Bill Gates sat there. So, but I think a lot of people like Jennifer Aniston.
Yeah. All right, we have some questions. Let's go to Michael.
Thank you very much, sir. Very interesting, very inspiring. That product that I mentioned earlier that you did for the gut bacteria and revitalization of gut health, is that available now on a commercial basis?
Not yet. We are talking about doing things like that, but all these things take a long time. We do have another company that's developing these kinds of things, actually in the nutrition area, called VitaKey, that's licensed some of our patents that the Gates Foundation funded. But products will take some time, probably.
Yeah, I understand. Thank you.
Sure.
Walter, you're muted.
Unmute myself there.
Yeah.
So Bob, a question first of all on the shampoos.
I'm glad I'm glad that I picked the right thing to talk about.
There you go. Do they contain PFAS?
What is it?
Do they contain PFAS? You mentioned Teflon, so PFAS is now considered a pollutant.
Yeah, originally it did. I think now they have ones that substitute for that, but that was kind of a template for that. So when that was first made back in 2006 or so, I believe that is what it contained. But I think now they have substitutes for it that have similar chemistries but not fluorinated.
Yep. And the other question is in regards to the cancer drug that's being developed by Moderna. Is that a treatment or a vaccine? And are there, and I know it's boutique for individual patients, is that going to be broadened into a more general or generic treatment or vaccine?
Yeah, well, first it is a vaccine, just to go over that for people. So, when you get something like COVID or flu, those are all what I'll call prophylactic vaccines. In other words, before you get the disease, I mean, you may know this, but before you get the disease, you get all these antibodies and immune cells. And if you get some foreign thing coming in, they'll attack it and hopefully destroy it before it can do anything bad to you. Of course, cancer is already starting to do something bad to you. So there has been the hope and the desire of what's called a therapeutic vaccine. And and so that's what this is. This is a vaccine, and it takes some of people's cancer where you're trying to know what it's composed of and make antibodies and sort of basically prime your immune system to attack it specifically. So you really can see that there are these what are called mutations or neoepitopes that are on the patient's cancer cells and yet not on their regular cells. So it's individualized, and then you would basically use that and inject it to treat each individual patient, and each vaccine would probably be different. You're right. I think that what this would be more analogous to, you probably have read about CAR T therapies, where you take the patient's cells and you get them to hopefully, work. But the hope is that someday, Moderna and Merck are in clinical trials for a number of cancers. BioNTech, which has sort of been a competitor of, which is good, I think, and Pfizer are also in clinical trials for different cancers. I mean, Moderna has gone after initially, like I say, melanoma. I think BioNTech is going after, they've done a smaller number of patients, but they've done pancreatic cancer, which is a very tough one, also with promising results. So I think what you'll see, again, none of these are approved yet. I think they will become approved, but for sure, if the data keeps up. But I think that it would be done. I mean, exactly the business model isn't necessarily clear to me, but I assume it would be something like what goes on with CAR T cells. That you would take the patient's cells and make the vaccine and train the body to and use it to train the body to attack the patient's cancer.
So again, these are individualized treatments. So is there anything being developed for a more generic treatment for individual cancers, but for the base population, so that you wouldn't need to extract the DNA from the patient before developing?
Well, I'm not sure. I think the question is, though, I think people have tried vaccines for cancer doing what you said. I don't think they've worked. I mean, I think the problem is that unless you really characterize what is different about the patient's cancer compared to the rest of your cells, I think then, from everything I've heard, I don't think you can. I don't think that it would work. People have tried for years to create cancer vaccines, and unfortunately, it hasn't. I don't think there's something generic about cancer that's just that you could do that way.
Okay, good. Thanks, Bob.
Yeah, and I'm not a cancer expert. But I think that what I said is right.
Sure. Okay.
Let's go to Goki.
Hi, can you hear me?
Yes.
Yeah. Hi. Hi, Professor Langer. This is a very inspiring story. Thank you so much. So here, you talk about a lot of non-viral delivery method. So I just wondering, do you have any opinion or any thought between, viral and non-viral delivery method here?
Well, I think what happens is, I think there's two different things. So people are using viral delivery methods for, say, DNA, and viral delivery methods, I would say in general, are more potent. They're more effective. They're also less safe. And I think the non-viral delivery methods, which, I talked about different lipid and polymer nanoparticles, they're generally not as potent, but they're safer. And certainly, I think for mRNA, clearly it makes more, or siRNA. I think it would probably make more sense to use non-viral. I think for DNA based, I mean, people have tried, including ourselves, to make non-viral, but they haven't been as potent. So right now, people are mostly using viral methods.
Oh, okay. Thank you.
Nanoviral vectors and things like that.
Yeah. So, the other question is, there's so many new area focused on the cancer treatment. For example, the cell therapy, gene therapy, antibody, bispecific antibody. So do you have any suggestion for our future cancer treatment?
I think that you'll see 3 or 4 new things, and you mentioned some of them. I think bispecific antibodies, CAR T cells, and cancer vaccines. I think those are all part of the future of new kinds of what I'll call general technologies. I think you'll continue to see new cancer drugs as well, including what I mentioned in the beginning, anti-angiogenesis drugs.
Okay. Okay. Yeah. Thank you. Finally, I talked to you in person. I worked for MIT, Brad Olson lab for many years before. We, but I never talked to you in person. Yeah. Thank you.
Sure. Well, give Brad my best. He's doing very good work.
Yeah.
Thank you.
Yeah.
Eddie.
Yes, well, first of all, Bob, fantastic work. Absolutely astonishing how a simple, well, not a simple, but an initial discovery essentially leads to so much. And I have to sympathize with your giving a talk at first time at a conference, because the first time I ever did was actually presenting data that completely contradicted the theory of the day. So turned out I was right, but I wasn't exactly pleasantly received either.
You can share that.
My question is, one of the things that's striking me about the biotechnology area is the convergence of multiple technologies, ranging from anything from AI to the development of nanotechnologies, 3D printing, and so forth. And I wondered if you could comment about how you see the technologies converging in the future, or what you can see coming down the pipeline. Because I have a feeling, in a sense, that biotechnology is in a sense converging like everything that developed into the iPhone and so forth. So what do you see actually happening in that future?
Oh, I think what you said is right. I think convergence. Phil Sharp and I actually wrote an article on that for Science, I think about 12 years ago. And I think some of the areas I talked about are really, I think, good examples of convergence, like tissue engineering, combining material science and cell biology. I think people mentioned artificial intelligence before. I think that's certainly, getting more and more attention in the biotech area. So I think that there are going to be a lot of things, and I think that, physical chemistry, there's this whole area of polycondensates that people are looking at that can affect, people like Phil Sharp and Richard Young and Arup Chakrabarty are working on areas like that. I think you're going to see, and we have companies like DuPont, which I've been on the advisory board of, along with the people I mentioned at MIT. So I think you'll see a lot of things along these lines.
One of the things that I was pondering about is the use of AI and the ability to do personalized production of vaccines, literally, almost using 3D printers and basic chemical synthesis. I sort of get the sense that is where we might be heading in the future. What do you think of that kind of progress?
I totally agree. I don't think there's just one direction. I think there are going to be many, and I think what you said is certainly one of them. But I think you'll see convergence, in many areas. 3D printing, like I was, giving that example with the microneedles, and we've done a number of other things too, which I didn't get into. But I think that's certainly. Like a lot of people are interested in what we call bioprinting for tissue engineering and so forth as well. So I think we'll certainly see that in the future. But I think, a lot of things, they'd almost be unexpected, things that you might, and I think that's been a good thing.
And one very quick last question, just because of my own ignorance, you might say. In your slide entitled Personal Cancer Vaccines, the last point, risk of recurrence of death reduced by 44%, with a P of 0.0266. I'm just curious, what does that P? I mean, it's a probability, obviously, but to me, usually when you give a good result, it's got a high P, not a low P. So I'm just curious about that.
I think it was, well, again, I thought, maybe I'm not sure I understand the statistics. I thought you wanted the P to be less than a certain number. In other words, I thought if it was less than 0.05, then it was hot statistically significant. Maybe I didn't make that as clear as it should be.
Yeah, I just wondered whether the P was on the pro or the or the negative side.
I see. So I probably should make that clear. I took it from the paper that they wrote, so I just
right. Just a point that there's just so much misinformation going around that somebody can easily clue in on that, and I've had problems exactly like that in the past.
I see.
So,
yeah, I mean, to me, if it was greater than 0.05, that's what I thought. You know, that then it would. But
then
this
it's a 95%
yeah.
probability.
So
that
this
it's
was
good.
less. This was
Yeah.
quite a bit less. I think. Yeah.
Yeah. So I think you should clarify that.
Yeah.
I just had so much misinformation. I've had
Pre-
stuff of my own
I
misused.
appreciate that. Thank you.
Yeah. Okay.
Let me just follow up on this conversation about the future and convergence with another question. Bob, advances in medicine have often been associated chronologically with waves. The first wave being Watson and Crick and the structure of DNA, and the second being the work on genomics. And I think now the third wave of advancement is in nanoscale science and engineering being introduced. But I just wonder if there might be a fourth wave. The convergence I'm thinking of is the convergence of very sophisticated computational methods with atom scale resolution experimental devices. I just wonder whether you think there might be a fourth wave associated with a convergence of that kind.
I think there very well could be. I don't know. I mean, there may be more than there may be a few more waves. But I think that what you're saying is certainly a giant area that people will do more and more.
Yeah, good. All right, let's go to Gunther.
Well, thank you for a great presentation. I mean, the breadth of your work is just stunning. I was particularly interested in the pulse release of antigens in the vaccines that you developed for applications in the third world. This will be a great game changer, no doubt. I was wondering, have you already tackled the stability issues that are typically associated with third world conditions like storage, climate, and so forth?
Yeah, well, that's one of the things we looked at in that Nature Biotech paper, that certainly the stability, I mean, it's not perfect, but I mean, we've got pretty good stability at 25 degrees. I mean, very good stability at 25 degrees for, 18 months. At 40 degrees, it wasn't as good, but it was certainly much better than, than if you just used regular liquid nanoparticles that were not formulated in microneedles. So I think but that's an area that we are working on, is how to stabilize it. And certainly in that Nature Biotech paper, it was stabilized a lot more than it's ever been before.
Angel.
Thank you so much for this great presentation. I, as a chemical engineer and also scientist, I would like to ask you if you could give us some advices about how to start a company with all your experience for the first time. And I'm sure that it was difficult, not the first company. If I would like to know about that.
Sure. Well, I'm no expert, but I mean, actually having, so for me, if you're at MIT, one of the nice things is the technology licensing office, that they, but what we've usually done is, like, I mean, I have students who are, in the venture capital industry, and they know what we're doing in the lab. And, I usually tell them, so say we're going to start a company, I might say to them, then you'd want to talk to now usually it's Lauren Foster in our office. You know, at one point it was Alita, and so they would talk to them about getting a license to the patent. But of course, there's other things too. You have to have a management team. You know, I mean, I'm a scientist. I'm not a business person, so you want to have a good management team. I think working in my experience, we've generally worked with you know, pretty experienced investors like at Polaris or Flagship, and they have done a very good job of building the company. Now they get to talk to a lot of people. They get to pick and choose who they'll work with and what ideas they work on. But I think having a good venture capital group or several good venture capital groups, that's a part of it. Acquiring the patents, and again, I don't know, you know, where you're located, you know, but if you're at MIT, you know, the technology licensing office is a great place to go. If you're not, you know, maybe you could file your own patents. But I think having intellectual property is certainly very important.
Okay, thank you so much.
Sure.
I'm always sensitive, Bob, to the time, and we're approaching noon. But I have Ron Smith is online in Toronto, and he has his hand up. So, Ron,
Okay.
Yeah, thank you, Ron.
We can't
Bob,
see you.
fascinating and illuminating. Thank you.
Well,
Ron,
thank you.
just for the record, we cannot see you. I'm not sure whether you're blocked or
Can you hear me?
can hear you. Yeah.
Yes, perfectly.
Okay.
Go ahead.
All right, so let's see whether or not that should
There
be
you go.
back now. Sorry about that. First of all, illuminating, Bob. Thank you so much for the information and the insights. This question is actually from, I guess, circularly Bill Gates, because it's from Microsoft Bing and it's chatbot. And so we asked it basically, if you had one question to ask of Professor Bob Langer of MIT and Moderna, what would it be?
Huh.
And we queried 9 different AIs to get the answer, and this one was actually the most interesting. So here's the answer that it came up with. If I had one question to ask him, it would be, how do you balance your roles as researcher, educator, mentor, and entrepreneur? Followed by, I would like to know how he manages his time, energy, and resources to pursue his diverse interests and goals. I would also like to learn from his experience and insights on how to foster a culture of innovation, collaboration, and excellence in science and engineering. Not a small menu, but here was the surprise. How may I be of service?
Oh, well, that's very nice. Well, let me that they're not the first ones to have asked those questions. And so what I would do, and not that I want to not answer them, except that it's not a short answer, but you know, so Nature, the journal, they followed me around. They wrote an article in 2009 to about a day in my life and how I managed my time. They were very interested in that. I had a lot of 15-minute meetings. I have a tremendous staff, but you know, I've pretty much always put the students first before I do anything else. And anyhow, so there's if you look under Being Bob Langer, I think that's what they ended up calling it. Not the most innovative title, but the woman Pearson, she did a very good job on the company thing and dealing with that. Harvard Business Review 2017 wrote a large article on a lot of those kinds of things and how I somehow combined everything as well. So those are 2 pretty good. They probably did a better job than I will do of explaining it. But you know, really, it's having great people who are, you know, independent, who you know, and it's working hard. And you know, I don't know what else to say.
Well, I guess the last one, just to follow up on the final exhortation, was how may I be of service? What do you see the usefulness of AI being in your pursuit in your fields?
Well, AI
How
will be
important or how unimportant would it be, do you think?
Well, I think AI is very important, but I don't think it addresses the things that I feel are most important, which are the personal aspects. I mean, the things that I'm proudest of really are how well my students have done, and you know, that is really trying to create an environment of what I'd call, you know, and I don't think you can do that just like, to me, what I've tried to do, and I learned this from Judah Folkman, is really create an environment where people feel that what they're doing is going to change the world, that it's going to make it better, more important, that they'll save lives, that they'll create things that people will enjoy, or products that will save lives. And so they're not just doing basic research. I mean, they are doing that, and they're publishing it in good places. But I think I have something like 10 of my students are professors at MIT, I think 10 at Stanford, 12 at Harvard, and I could go on. I mean, I have 400, and a lot of people have started companies. And I don't know that AI can do that. AI, I think, is great and will help us, and it already is helping us on things like, well, how can we make better nanoparticles? Because you can take a lot of data and make better nanoparticles if you understand what chemical structures allow you to do certain things and not others. But I don't think that the company thing I mean, maybe once you have the company started, I could see where AI will be very useful in intellectual property. So I do think that there can be a lot of places that it can have the role, but I don't know how it trains somebody to be a person who's going to be a leader. That's not clear to me yet.
It's going to take time to know, isn't it?
Yes. Well, and I think people keep trying. I mean, the questions that people have asked me about how I do things, I mean, I've had a lot of people look at that, and I think about that for other people that I've known. I don't know how you can predict in advance, what kind of people will be good at what and not others. I mean, I've certainly thought about that myself. It's a great question.
Bob, I'd like to bring the session to a close and thank you for this wonderful presentation. My opinion is that science and technology at its best serves a societal purpose, a useful societal purpose. But this is not always the case. And, what comes to mind immediately is social media, which I think has served us poorly. And that's just my opinion. Maybe others would agree or disagree. But Bob, what you've given us today are many examples of societal benefit from your work. And more than that, you leave us today with the hope of more to come. I want to thank you for that. The presentation was wonderful. Your message was inspirational, and the humanity of it was even more remarkable. I hope that the young people who will see this recording, the kids who are in class today, right now, unfortunately, but when they see this and listen to your words, will be moved by them, as I think we are. Bob, once again, thank you very, very much for joining us. I'm very, very appreciative of your being here. I want to thank you on behalf of all of us.
My great pleasure. Thank you all. Great questions. And it's an honor and a pleasure. Thank you.